Liver disease is a type of damage to or disease of the liver. Whenever the course of the problem lasts long, chronic liver disease ensues. Some of the signs and symptoms of liver disease are the following. There are more than 100 different kinds of liver disease. These are some of the most common. Liver disease can occur through several mechanisms. One general mechanism, increased DNA damage, is shared by some of the major causes of liver disease. These major causes include infection by hepatitis B virus or hepatitis C virus, alcohol abuse, and obesity. Viral infection by the increase in intracellular ROS is about 10,000-fold with chronic HBV infection and 100,000-fold following HCV infection. This increase in ROS causes inflammation and further increase in ROS. ROS cause more than 20 types of DNA damage. Oxidative DNA damage is mutagenic and also causes epigenetic alterations at the sites of DNA repair. Epigenetic alterations and mutations affect the cellular machinery that may cause the cell to replicate at a higher rate or result in the cell avoiding apoptosis, and thus contribute to liver disease. By the time accumulating epigenetic and mutational changes eventually cause hepatocellular carcinoma. Epigenetic alterations appear to have an even larger role in carcinogenesis than mutations. Only one gene, TP53, is mutated in more than 20% of liver cancers, while 41 genes each have hypermethylated promoters in more than 20% of liver cancers. Alcohol consumption in excess causes a buildup of acetaldehyde. Acetaldehyde and free radicals generated by metabolizing alcohol induce DNA damage and oxidative stress. In addition, activation of neutrophils in alcoholic liver disease contributes to the pathogenesis of hepatocellular damage by releasing reactive oxygen species. The level of oxidative stress and acetaldehyde-induced DNA adducts due to alcohol consumption does not appear sufficient to cause increased mutagenesis. However, as reviewed by Nishida et al., alcohol exposure, causing oxidative DNA damage, can result in epigenetic alterations at the sites of DNA repair. Alcohol-induced epigenetic alterations of gene expression appear to lead to liver injury and ultimately carcinoma. Obesity is associated with higher risk of primary liver cancer. As shown with mice, obese mice are prone to liver cancer, likely due to two factors. Obese mice have increased pro-inflammatory cytokines. Obese mice also have higher levels of deoxycholic acid, a product of bile acid alteration by certain gut microbes, and these microbes are increased with obesity. The excess DCA causes DNA damage and inflammation in the liver, which, in turn, can lead to liver cancer. A common form of liver disease is viral infection. Viral hepatitis such as hepatitis B virus and hepatitis C virus can be vertically transmitted during birth via contact with infected blood. According to a 2012 NICE publication, about 85% of hepatitis B infections in newborns become chronic. In occult cases, hepatitis B virus is present by HBV DNA, but testing for HBs AG is negative. High consumption of alcohol can lead to several forms of liver disease including alcoholic hepatitis, alcoholic fatty liver disease, cirrhosis, and liver cancer. In the earlier stages of alcoholic liver disease, fat builds up in the liver cells due to increased creation of triglycerides and fatty acids and a decreased ability to break down fatty acids. Progression of the disease can lead to liver inflammation from the excess fat in the liver. Scarring in the liver often occurs as the body attempts to heal, and extensive scarring can lead to the development of cirrhosis in more advanced stages of the disease. Approximately 3 to 10% of individuals with cirrhosis develop a form of liver cancer known as hepatocellular carcinoma. According to Tilg et al., gut microbiome could very well have an effect, be involved in the pathophysiology, on the various types of liver disease which an individual may encounter. Particulate matter or carbon black are common pollutants. The following factors are the harmful effects of liver exposure under PM or CB. First, they have an obvious direct toxic effect on the liver. Chemicals will affect metabolism and impact liver function. Second, inflammation of liver caused by PM and CB impact lipid metabolism and fatty liver disease. Third, PM and CB can translocate from lung to liver. PM and CB can easily translocate from lung to liver because they are very diverse and each has different toxicodynamics. Detailed mechanisms are not clear. Water-soluble fractions of PM is the most important part for PM translocation to liver through extrapulmonary circulation. When PM goes through blood vessel into blood, it combines with immune cells that will stimulate innate immune responses. Pro-inflammatory cytokines will be released and cause disease progression. A number of liver function tests are available to test the proper function of the liver. These tests for